Hello and welcome. I'm Dr. Mark Brandon, an associate professor at the School of Medicine and the Department of Psychiatry at the Douglas Hospital Research Center at McGill University. As a neuroscientist, my research focuses on understanding how brains navigate and comprehend the world around us. Today, I'm thrilled to present another installment of our publication series. This platform allows me to share groundbreaking neuroscience research directly with you, whether you're a fellow scientist or simply fascinated by the brain. My goal is to make the complex world of brain science both accessible and engaging. This episode highlights a new study from our lab, published today in Cell Reports, titled Optogenetic Silencing of Medial Septum GABAergic Neurons Disrupts Grid Cell Spatial and Temporal Coding in the Medial Entorhinal Cortex, led by Dr. Jennifer Robinson, an extremely talented postdoctoral researcher in my laboratory. This work was done in collaboration with Dr. Michael Hasselmo at Boston University. Our study focused on grid cells in the medial entorhinal cortex, which are essential for spatial navigation. Grid cells fire in a repeating hexagonal pattern, helping to form a spatial cognitive map that aids in navigation. Importantly, our previous work has shown that these cells are the first functional cell type to be disrupted in Alzheimer's disease. This hexagonal spatial pattern of firing locations produced by grid cells heavily relies on inputs from the medial septum. During my graduate work at Boston University in Dr. Mike Hasselmo's lab, we showed that completely inactivating the medial septum disrupted the spatial firing pattern of these grid cells. Instead of firing in a hexagonal pattern, grid cells fired all over the environment, losing their spatial tuning. However, the story doesn't end there. The medial septum contains three types of neurons: GABAergic, cholinergic, and glutamatergic. In our current study, we aim to identify which cell population is responsible for the disruption of grid cells. My graduate work inactivated all medial septal cell types, so we knew that the medial septum was essential, but we didn't know which subpopulation was responsible for the disruption. In our new study published today, we focused on the GABAergic cells in the medial septum. We targeted these cells because previous work has shown they are critical for establishing the theta rhythm in the hippocampus and entorhinal cortex. The theta rhythm is a large neural oscillation that coordinates the activity of nearly every neuron in these regions. To investigate the role of medial septum GABAergic neurons, we used optogenetics to selectively inhibit them during grid cell recordings. This cutting-edge technique allowed us to precisely control neural activity and observe the effects on grid cell function. Let me briefly introduce you to these optogenetic methods. We used the VGAT-CRE transgenic mouse line, which expresses CRE recombinase in all GABAergic neurons in the brain. We then injected a CRE-dependent adeno-associated virus into the medial septum. This virus is designed to express a protein of interest only in the presence of CRE recombinase, therefore achieving cell type selectivity. In this case, our virus expressed a protein called ARCH-T, a light-sensitive outward proton pump that allows for powerful, temporary, and repeatable silencing in response to brief light exposures. To introduce light to these cells, we implanted a fiber optic into the medial septum and used an implant for electrical recordings of neurons in the medial entorhinal cortex to record from grid cells. Now, on to our findings. Inhibiting medial septum GABAergic neurons with our optogenetic approach significantly disrupted the spatial periodicity of grid cells. This demonstrates that the GABAergic subpopulation in the medial septum is crucial for supporting the normal activity of entorhinal grid cells. In addition to spatial coding, we examined the temporal coding of grid cells, specifically looking at theta phase precession. Theta phase precession is a phenomenon where the timing of grid cell firing relative to the theta rhythm encodes information about the animal's position in space. Our results showed a significant attenuation of theta phase precession during the inhibition of medial septum GABAergic neurons, indicating that these neurons are crucial for both spatial and temporal aspects of grid cell coding. Interestingly, we also observed that while theta oscillations returned immediately after the inhibition period ended, grid cell spatial coding took longer to recover. This suggests that the network dynamics underlying grid cell function require more time to stabilize following disruptions to theta oscillations and inactivation of medial septum GABAergic inputs. These insights are crucial for understanding not only basic navigational processes, but also the broader implications for neurological conditions where navigation and spatial memory are compromised. Our previous work has shown that grid cells are the first cell type to show disruption in Alzheimer's disease. Our findings here highlight the importance of medial septum GABAergic neurons in maintaining the integrity of the grid cell network, suggesting that the medial septum GABAergic to entorhinal circuit may degenerate early in Alzheimer's disease. We can't wait to put these ideas to the test. Thank you for joining us today as we explored the critical role of medial septum GABAergic neurons in grid cell function and their importance for spatial coding and memory. Stay tuned for more episodes where we'll continue to unravel the mysteries of the brain's navigation and memory systems.